Hello, and thanks for joining us today for the latest Technology Networks webinar, Characterization and Sequence Mapping of mRNA Therapeutics using Mass Spectrometry. I'm Kate Robinson, Editorial Assistant for Technology Networks, and I'm here to moderate the event. I'm pleased to have Professor Mark Dickman joining us today as your presenter. Mark obtained a first-class honours degree in biochemistry and chemistry at the University of Sheffield. He continued at the Krebs Institute, where he obtained his PhD. Following his PhD, Professor Dickman joined a biotechnology company, Transgenomic Limited, where he worked as a research scientist developing analytical techniques including DNA and RNA chromatography. He is currently a professor in bioanalytical science and engineering in the Department of Chemical and Biological Engineering at the University of Sheffield. A warm welcome to you, Mark. Before and after today's presentation, we will be asking a poll question, for which we would appreciate your responses. The first poll question is, what technology do you use to confirm mRNA sequence? Is it next-generation sequencing, Sanger sequencing, LC-MS, other, or not working on mRNA currently, but plan to start soon? Please submit your answers now. Following the webinar, we will have a Q&A session, and we welcome any questions that you may have. You can submit questions for the Q&A session at any time during the presentation. To ask a question, you should enter it into the box on the right-hand side of your screen and click Send. We will answer as many as possible during the time available. For any questions that we don't have time to get to, we will be sure you are contacted offline with an answer. If you experience any technical issues, please click the chat icon in the bottom right corner to request support. Please remember, you can ask questions at any time, even whilst watching the webinar on demand. So, without further ado, I will now hand over to Mark.
Thank you very much for the introduction, and thank you very much to the organisers for giving me the opportunity today to give a presentation on some of our recent research that we've been performing at the University of Sheffield, developing novel analytical tools for the characterization of mRNA therapeutics. So by way of introduction, I'm just going to outline what I'm going to be presenting here today. I'm first going to give you a bit of an introduction into RNA therapeutics, discuss the importance of this new class of drug modality, and then I'll start to move on to discuss some of the analytical challenges of analysing these long RNA and mRNA therapeutics. I'll then move on to some of the descriptions of some of the new and novel workflows we've developed here in Sheffield, using RNase mass mapping and liquid chromatography mass spectrometry workflows to provide further insight and characterization of this important class of molecule. So first, by way of introduction, a lot of this talk and a lot of our work we do in Sheffield is based on analysing RNA. So this slide just shows you a schematic illustration of the backbone of RNA, which contains a phosphodiester backbone, which is negatively charged. So RNA is a polyanion, and you can imagine it's a negatively charged macromolecule. So not only does it have a phosphodiester backbone that's negatively charged, connected to this phosphodiester backbone are ribose molecules, and then to each of these sugar molecules, we have the different 4 bases, C, G, A, and U. So looking at the structure of RNA and nucleic acids, obviously everyone's probably familiar with the double helical structure of DNA, which is shown here on the left-hand side of the slide. But RNA adopts much more different and complex structures. So typically RNA is a single-stranded molecule. But when it comes to analysing or looking at these really large RNA molecules that are present both in nature and are part of these large mRNA therapeutics, these really large RNA molecules can adopt really complex secondary and tertiary structures, which is depicted here on the right-hand side of the slide. So they can contain large single-stranded regions, but also regions where we have duplex or complementary structures. So if you think of long RNA or these mRNA therapeutics, there's a really long piece of string that can kind of fold up into form into lots of complex shapes and structures. So we've been working here in Sheffield quite a number of years now, sort of analysing these different RNA therapeutics, and they're really emerging as a very important class of drug modality or drug product. And some of the areas in which we've been working on here in Sheffield are kind of 3 different classes of different therapeutics or nucleic acids. So we have here the oligonucleotide therapeutics shown on the left-hand side of the slide. So these are the traditional smaller nucleic acids, typically about 20 to 30 nucleotides in length. So these are the smaller oligotherapeutics, which can be antisense oligonucleotides or double-stranded siRNA molecules. Now, these work by a mechanism of RNA interference, and there are a number of classes emerging or a number of drug products now emerging in this area. Now, on the other hand, we have these larger mRNA therapeutics. So these are now much larger molecules. So these are the ones about over 1,000 nucleotides in length. So these are much bigger compared to the traditional oligotherapeutics. And obviously, everyone's kind of aware of the area of mRNA therapeutics that's just really recently taken off by the presence or the production of 2 mRNA vaccines that have been approved for COVID-19, manufactured by Pfizer-BioNTech and Moderna. And there's also a host of many other different mRNA vaccines under development for many different companies in the pipeline. So the aspects of this talk, what I'm going to do is focus on this area, this class of molecule, these mRNA therapeutics, these large RNA molecules, which have been used as vaccines for COVID-19. So it's a really growing area, significant demand. There's a big pipeline of these molecules in production. And some recent research has shown currently in last year, in 2021, the market share for mRNA therapeutics is about 46.7 billion. And it's expected to grow to about over $100 billion by 2026. So it's a real key emerging area for the biopharma and biotech industry. So I'm going to describe a little bit of how these mRNA therapeutics work. They essentially follow the central dogma of molecular biology. And this is where we have the DNA, which is transcribed into mRNA, and that mRNA is then translated into the protein. So these mRNA therapeutics essentially fit here in the middle, where we have the mRNA sequence, and that's translated into a protein. So it's used to make proteins, essentially. So these can be used as vaccines, as we're all well aware, the ones that are currently in development for COVID-19 and being used for COVID-19. They can also be used as protein replacement therapeutics and also in the areas of regenerative medicine. So how do these molecules work? How do these typical mRNA molecules work as vaccines or therapeutics? So on this bottom slide is a bit of a schematic diagram showing you in principle how these molecules essentially work in the body. So on the left-hand side here, we have our mRNA therapeutic. So this is essentially your sequence of RNA or mRNA. Now we have to get that into the cell. That's the first thing for these to work. So we essentially package these by surrounding them by a lipid nanoparticle. So essentially we have here in blue, the mRNA molecule surrounded by a lipid layer. And this enables the mRNA to enter the cell. So once it's inside the cell, essentially the mRNA just hijacks the cellular machinery that's used to make proteins. So as it's released from the particles, the naked mRNA is essentially hijacks the machinery, which is then used within our cells to make lots of protein. So within the sequence of the mRNA, we can use any sequence we want. We can define and engineer the mRNA molecule to make our protein that we desire. So in the case of mRNA vaccines that are used in the COVID-19 treatment, we essentially have the mRNA sequence encoding for the spike protein. So once the RNA is inside the cell, essentially it hijacks the cellular machinery and we make lots of copy of the spike glycoprotein. So once this protein here, shown here in purple, is made inside the cells, we make lots and lots of this particular protein. This then presents itself on the surface of our cells, and then this elicits an immune response and we start to raise antibodies against that protein. So this is the overall concept in which how mRNA vaccines or mRNA therapeutic process works. Essentially, it's a way of using or generating an RNA sequence, encodes a protein, and then that protein elicits an immune response in the cell and we make antibodies against that particular protein. So how are these manufactured? So the actual manufacturing process is fairly straightforward. It's done in vitro, so essentially it's done inside a test tube. It doesn't require any cells or cell factories to make this. So it's made by a mechanism using enzymatic synthesis, and this is a process called in vitro transcription. So in vitro transcription, essentially we take our plasmid DNA template. So this is the DNA sequence that essentially encodes the RNA that we need to synthesize. We then add an enzyme called T7 RNA polymerase, the building blocks that are required to make the RNA molecule called nucleotide triphosphates. We put all those together in vitro in a test tube, and this makes our mRNA product. And just shown here on the slide is a schematic illustration of what a typical mRNA therapeutic has. We have a cap on the 5' end, followed by a 5' UTR sequence. And then we have the main sequence of the RNA, and this is the open reading frame. And this sequence corresponds to the gene of interest or the protein that we want to make. So you can design the sequence, in particular, any sequence to essentially make any protein that you wish to make inside the cells. After the gene or the open reading frame, we have a 3' UTR region, typically followed by a 3' poly A tail. This is usually typically about 100 to 120 nucleotides in length. So it's essentially made via in vitro transcription, requires an enzyme, the building blocks, optimization of the process with the DNA template, and we essentially manufacture our mRNA. But of course, following the manufacturing process, there's lots of contaminants in the material that we've made, such as the enzyme, the T7 polymerase, unused NTPs. We've got the plasmid DNA template left over. We've also got things like failure sequences, degradation products, incomplete capping, incomplete polyadenylation products, and also the presence of double-stranded RNA, which is an unwanted molecule in the mixture. So post-synthesis, there's lots of impurities that we need to remove prior to using this particular mRNA. So this is where the field of analytics comes into play. There's a real demand to develop analytical methods to really purify and analyze the mRNA therapeutics, because these are really critical, first and foremost, to underpin manufacturing development. Obviously, the manufacturing of large amounts of these vaccines at scale is a big problem. It's a kind of current bottleneck. So any of the manufacturing development opportunities where we want to increase the yield, we want to increase the quality of the product, then it's really key and important to have really good, robust analytics where we can measure how much mRNA we're making and the quality of the mRNA that's being produced. So analytics plays an important role in manufacturing development. Obviously, analyzing the mRNA plays an important role at looking at batch-to-batch manufacture and process repeatability, where we again need to measure the yield and the quality of the RNA from batch-to-batch manufacturing processes. And then finally, the analytics plays a really key role in looking at the quality of the mRNA produced. So we need to measure things like mRNA critical quality attributes, CQAs, looking at the purity of the molecule, the amount of degradation, etc., how much of the molecule has the 5' cap, etc. So currently, there are very limited or few analytical methods that are able to characterize really large RNA molecules. So the analysis of large RNA molecules over 1,000 nucleotides in length is particularly challenging. And there are not many robust, high-throughput analytical methods out there that can really accurately analyze these class of molecules. So there's significant demand for the development of new and improved methods to characterize large RNA molecules, including mRNA therapeutics. So what I'm going to do now is move on to some of the methods we've been developing here at the University of Sheffield to really start to develop new methods for the analysis of some mRNA therapeutics. And I'm going to talk you through some of the analysis that we've done producing some of the mRNA vaccines or mRNA therapeutics against the SARS-CoV-2 spike protein. So some initial work that we did a number of years ago now during the pandemic here in the University of Sheffield, a collaborative venture with Professor David James and the NHS Trust here in Sheffield, where we developed a platform for the production of the large amounts of the spike protein for serological assays in the NHS Trust here in Sheffield. So we had the constructs in place, the plasmid DNA templates to produce the spike protein. So we simply switched from making the in mammalian cells to now making the mRNA using in vitro transcription. So we have our plasmid DNA template encoding the sequence for the spike glycoprotein. So we essentially incorporated that in vitro, put it into an in vitro reaction with the polymerase, the NTPs, and this was then able to manufacture our 3,900 nucleotide mRNA corresponding to the spike glycoprotein. So post-purification of the larger mRNA, we were able to carry out ion pair reverse phase HPLC to analyze the quality and the purity of the material. So this is a nice example showing that we can use HPLC to give us important information on the yield and the quality of the material. And we've done lots of development over the last 10 or 20 years, developing and optimizing this class of chromatography to analyze these large RNA molecules. But what the chromatography doesn't tell you is any information about the sequence or confirmation of the sequence of that particular molecule. So whilst we can get information on the yield and some information on the quality, we need to start thinking about alternative analytical methods to look at the sequence and sort of identify or corroborate the identification of the sequence of your mRNA therapeutic. So this is where an alternative technique comes into play, and this is called RNase mass mapping. So RNase mass mapping works essentially by taking a really large RNA molecule and chopping it up into much smaller, defined, discrete oligonucleotides using an enzyme such as RNase A or RNase T1. So these particular enzymes will cleave after certain bases in the RNA sequence, generating a subset of smaller oligonucleotides, and then we simply separate those out and analyze them, sequence them and identify them in the mass spectrometer. So we've used RNase mass mapping over the number of years for a wide variety of different applications quite successfully, including the analysis of the CRISPR-Cas systems, looking at CRISPR RNA processing, and also quantifying and analyzing double-stranded RNA biopesticides. However, one of the big problems of using this technique to analyze really large RNA is it gives us lots of particular problems. So first and foremost, while RNase mass mapping works quite nicely for small RNAs, when it comes to analyzing really large RNA molecules, such as these mRNA therapeutics, the problem with these high-frequency RNAs, such as RNase A and RNase T1, is that it chops it up into too many smaller fragments. Okay, this results in lots of small oligonucleotides that map to multiple points along the RNA. It doesn't give us much useful information. Also, many of the masses or many of the oligonucleotides are sequence isomers, which again gives us many problems trying to do sequence mapping. The other problem of analyzing these really large RNA molecules is the lack of robust software to handle really large data sets. So when we chop these really large mRNA therapeutics up into fragments, it generates huge numbers of fragments and very complex mass spectrometry data sets. And what we really need is kind of robust software, high throughput software that can manage, interpret and identify this class of oligonucleotide fragments. So I'm now going to move on to the description of the workflow that we've now developed here at the University of Sheffield for the analysis of the mRNA therapeutics. This is an outline of essentially the workflow that we've developed. I'm going to talk you through step by step each one of the different components of the workflow, and then spend a little bit of time going into detail of how we developed each one and put this together in a complete package. So the workflow that we developed here for the development of the analysis of the mRNA therapeutics, the first stage is essentially the partial T1 digestion of your mRNA therapeutic. So essentially we have RNase T1 immobilized on magnetic beads. And what we do is a partial T1 digest of our mRNA therapeutic by simply incubating the mRNA with the immobilized beads for a very short period of time. We then essentially remove the magnetic beads, and this allows us to generate a partial T1 digest, generating large oligonucleotide fragments with multiple miscleavages. The next stage is essentially the high-resolution separation of the oligonucleotide fragments. So this has been achieved using the thermoscientific DNA pack RP columns in conjunction with ion pair reverse phase chromatography. And the ion pair reverse phase chromatography was performed using the thermoscientific Vanquish UHPLC system to give us this high resolution separations prior to the mass spectrometry analysis. So all the mass spectrometry analysis was performed using the thermoscientific Orbitrap Exploris 240 instrument. And then all the data analysis was performed using the Thermo Scientific Biopharma Finder software. So we essentially integrated this workflow through from partial T1 digestions, high resolution ion pair reverse phase chromatography, mass spectrometry, and data analysis. And what I'll do is in the preceding slides is talk you through each one of those step by step. So the first step is the RNAs digestions. So I said before, here's a schematic illustration of our mRNA therapeutic with our 5' cap, the UTR, the coding sequence, the 3' UTR, and the poly A tail. Now, under traditional RNase digestions using a complete RNase T1 digest, this essentially chops this RNA molecule up into too many short fragments. So here on the right-hand side, I've shown schematically, hopefully, that we get many different fragments that are all the same. So these are non-unique fragments. They map to multiple points across the mRNA, and they're not really very useful for sequence mapping. So the approach we've taken is to take the mRNA therapeutic, do a partial RNase T1 digestion using the RNase T1 on the magnetic beads. This then gives us the opportunity to control the reaction. And essentially, in this case, what we do is we generate much larger fragments with multiple miscleavages. And these larger fragments then map to unique sequences on the mRNA molecule. So by using this approach, we get much more higher sequence coverage in principle. So these RNase digestions are essentially performed using RNase T1 and A immobilized on magnetic beads using the Thermo Scientific Smart Digest kits. And we've been able to adopt this in an automated fashion by incorporating these digests on the Thermo Scientific Kingfisher automated robotic system. So we can do this in a fully automated fashion. So this is an example of some of the data that we get, showing some of the chromatograms, showing some of the digests. So it's very easy to control the digests by simply either changing the incubation time or the amount of enzyme that we use in the reaction. So here's 3 different chromatograms. We have here the series of different oligonucleotide fragments. Small fragments elute here early on the left-hand side, where the large oligo fragments are shown here on the right-hand side. Now, if we increase the amount of RNase T1, then what we'll get is a reduction in the amount of these larger oligonucleotide fragments as it moves towards more of a complete RNase T1 digestion. So essentially very easy to monitor these digestions, and it's simple to control the reaction by either changing the amount of the enzyme or the time course of the reaction. So we were also able to show that we get also very reproducible RNase T1 digestions by doing it all on the automated digestion platform. Here's an example showing using EGFP mRNA by using exactly the same amount of beads for the same amount of time across 3 different biological replicates. You can see we get really good reproducible chromatograms demonstrating we get reproducible cleavage of our mRNA using this approach. So the next step, once we've done the digest, is performing the ion pair reverse phase chromatography. So this was all performed using a macroporous polymeric column, the DNA pack RP column. So the advantages of this particular column, it's stable at high pH and stable at high temperature, which is really beneficial for the ion pair reverse phase chromatography. So we typically perform the reaction or the chromatography using TA in HFIP, and typically the column temperature up at about 50 degrees C. So you can see here we have an oligonucleotide standard. We get very high resolution separations from small oligonucleotides down at 5 nucleotides in length, all the way up to about 36 or 40 nucleotides in about a 40-minute window. So by using this ion pair reverse phase chromatography, analyzing a typical RNase T1 digest, this is the type of chromatography we get. We get high resolution separation of a very complex series of oligonucleotide fragments. So I said also before, it's not just the digests, the separations and the mass spectrometry analysis, but the data analysis is also really key and important. So we generate these really large data sets, complex series of oligo fragments. So what we need is robust, important tools that can do the data analysis. So all this was done in Biopharma Finder. And this allows us to use the sequence manager tool option, where we can put in our sequence or our RNA sequence of interest that corresponds to the mRNA therapeutic molecule. We essentially take the sequence, put it in there. It allows you to put in any modifications of interest. And essentially what it does, using the RNase mapping option within the software, we can select the enzyme that we've chosen to use in our process, essentially combine it with the mRNA sequence of the mRNA therapeutic you're working on. And then essentially what it does, based on both the high resolution MS1 data and the tandem MS2 data from the sequencing of the oligonucleotide fragments, it essentially shows you all the oligonucleotides that have been identified that match your mRNA sequence. And then it also generates a powerful display platform by showing your overall sequence coverage against your mRNA therapeutic. So it offers automated analysis in conjunction with powerful data visualization tools for the sequence mapping. So this is an example of some of the data that we get out of the Biopharma Finder software once we've done the identification. So here's one of the typical base peak chromatogram. And I've just selected some of the identifications of some of the oligonucleotide fragments. And you can see typically here on the left-hand side of the early retention time, we see the smaller oligonucleotide fragments. And as we move towards the right-hand side, the later retention time, we now see the identification of these much larger fragments with many miscleavages. Also within the Biopharma Finder software, and using this approach, it also does allow us to identify sequence isomers. So here's an example of two different sequence isomers that have been identified. So sequence isomers essentially are the same mass, identical mass, but they have different base sequences. So here's an example. Even with using a partial digest, sometimes we generate a number of these sequence isomers, but under the chromatographic conditions, these are typically separated. And this allows us then to identify these different sequence isomers with identical masses. And this is the data output from the Biopharma Finder, showing the tandem mass spectrometry information, the fragment ions corresponding to the two different sequence isomers, enabling us to identify and discriminate between the two. This is a further example. I'm just showing here two slightly larger oligonucleotides, again the same monoisotopic mass. We get separation in the chromatography, which based on their fragmentation allows us to identify the two different sequence isomers. The other advantage is taking advantage of the kind of the speed and the sensitivity of the mass spectrometry workflow. A typical oligonucleotide is shown here with the charge state distribution under the ion pair reverse phase conditions that we use in our chromatography. So what we typically see is our oligonucleotide fragment, and we see ions across different charge states, shown here as the 4-, the 5, the 6, 7, 8, and 9. And using the speed of the Exploris 240 instrument, it's possible to obtain MS2 fragmentation across all the different charge states, and this gives us extra confidence in the identification of our different oligonucleotides. So here's some examples that show that we get fragmentation, tandem MS information at the 4- charge state, the 5-, 7-, and the 8-. So this really builds our confidence by identifying the oligonucleotide across multiple different charge states. Okay, so I've gone through the workflow kind of step by step. And what I'm going to do now is show you some of the examples of how we've applied this workflow for sequence mapping of some mRNA therapeutics. So I've got two examples here shown on the left-hand side of the slide, where we produced and purified some mRNA corresponding to the SARS-CoV-2 spike protein mRNA and a control EGFP mRNA. So we essentially take these two mRNA molecules, we perform our partial T1 digest and our workflow that we've described. Here shows the ion pair reverse phase mass spectrometry analysis, showing the two TICs of this. And then when we put this through the data analysis and then the sequence mapping. So what we wanted to do is use this type of workflow and compare it against a traditional RNase mass mapping, doing a complete T1 digest using the same data analysis software. So shown here is the sequence coverage differences that we get when we compare the analysis of both the EGFP mRNA and the SARS-CoV-2 spike protein mRNA against a complete T1 digest, against the new method that we've developed here using a partial T1 digest. So you can see here we get a much more significant increase in sequence coverage typically about 84-85% sequence coverage of the partial T1 digest, compared to the traditional RNase mapping using a complete T1 digest, where we're typically only getting very limited sequence coverage, typically 11% and 24% for the EGFP and the SARS-CoV-2 spike protein. So you can see we've achieved a much increase in the sequence coverage, significant increase in the sequence coverage, allowing us to fully characterize in detail, confidently assign the mRNA therapeutic in this case. In addition, it's important also in our data analysis to take our data analysis software files and also make sure that we're getting very little matches against a random sequence. So in this case, what we do is essentially we generate a random sequence of the same size and the same GC content as our mRNA therapeutic. And then we search our MS data against that and look for the number of oligonucleotide matches. And you can see in both cases, using the workflow and the data analysis tools that we've developed and used, we get little or only very small amounts, less than 1% matching against the random sequence. Again, giving us good confidence and validation of this method in our sequence characterization. The software also allows us to look at the overall sequence coverage patterns. And here's two examples showing the difference in the sequence coverage patterns that we get comparing the complete T1 digest against our newly developed partial T1 digest method. So the identified fragments are shown in green and the non-identified fragments are shown in red. So again, you can see we're getting significantly improved sequence coverage. Those of you with eagle eyes might notice at the end of the EGFP mRNA, we have a kind of a block of red here for not identified. And this corresponds to this large poly A tail, typically about 100 to 120 nucleotides at the end of the mRNA molecule. And using this approach or the developed technology used in this assay, it's unexpected that we would not find this particular molecule because it's too big to essentially sequence and identify in this method. So once we developed these applications to look at some of these mRNA therapeutics, we thought we'd look at some other larger RNA molecules as an example of a more challenging kind of class of molecule. So we looked at some alternative RNA molecules, such as the MS2 RNA, which is about a 3,659 molecule. And this we thought was a particularly challenging molecule to analyze using this particular workflow, mainly because it has a high degree of secondary structure. So it has lots of duplex regions within the RNA structure. Now, RNase T1 essentially, under the conditions, will only cleave single-stranded RNA. So we thought this would be a good example as of a very difficult molecule to analyze under partial T1 digest conditions, where the enzyme's only used for a short period of time to generate oligonucleotide fragments. So what we did was take this particular molecule, put it through the same workflow and the data analysis, and then again look at the sequence coverage that we get. So what we found was by altering the conditions and just putting in different amounts of enzyme for different amounts of time, we were again able to obtain significantly higher good sequence coverage of over 70% for this particular challenging molecule. And again, the complete T1 digest was only typically able to identify about 20% sequence coverage. So again, we've been able to optimize, and for even particularly challenging large RNA molecules with high degrees of secondary structure, we can still obtain really high sequence coverage. So one of the other areas we've extended this analysis to, and that's using modified nucleotide triphosphates as well. So many of the mRNA vaccines used for COVID-19 contain a modification, and this is this N1-methyl pseudouridine. So during the manufacturing process, essentially N1-methyl pseudouridine triphosphate is used to replace UTP. So all the uridine molecules become N1-methyl pseudouridine. So essentially what we did was then synthesize some mRNA containing the chemically modified nucleotide triphosphate and put it through essentially our workflow. And all it requires is to essentially, within the data analysis or within Biopharma Finder software, we essentially just modify our sequence. And instead of having uridines, we replace all the uridines with the N1-methyl pseudouridine. So here's two examples of where we analyzed some mRNA that was both contain the chemical modification and the absence of the chemical modification. In this case, we looked at 5-methoxy-U. So we had FLUC mRNA with 5-methoxy-U in there. We put it through our developed workflow, did the partial T1 digest, mass spectrometry analysis, and searched against either the mRNA sequence corresponding to either just uridines or the 5-methoxy-uridines. And you can see here in both cases, we were able to obtain over 90% sequence coverage for the open reading frame for the FLUC mRNA, showing the ability that we can detect the chemical modifications. We also did some further analysis, some further validation by looking at some of the specific oligonucleotide fragments with and without the chemical modifications, and it allows us to accurately verify the sites of a number of the different modifications shown here in red in the fragmentation pattern. So in the final examples of this workflow and this technology, we looked at extending this to analyze RNA impurities within the RNA samples. So what we did in this case as an example, as a proof of principle, we essentially took the SARS-CoV-2 spike protein mRNA, and then we mixed it with varying mass ratios of some ribosomal RNA, which was used to represent some potential impurities in our RNA product. So we combined the two together, performed the partial RNase digest and the mass spectrometry analysis, and then looked to identify these known RNA contaminants or known RNA sequences. So you can see here we mixed at two different mass ratios, 100:1 and 10:1. And you can see here at the 10:1 ratio, we were able to identify the ribosomal RNA representing our RNA contaminants in this case. So again, the power for this approach is we can start to look for potentially known RNA contaminants that we may want to be able to identify. Or also this is an opportunity where we can look at multiple RNAs. So in this case, might be an example where we have multiple mRNA vaccines in the same sample. So multiple different mRNA sequences. We can put through this sequence mapping approach. And again, we can identify multiple RNAs within the same mixture or the same sample. So just in conclusion, now I'd like to summarize some of the workflows and some of the work that we've done. So we've developed a fully automated LC-MS workflow for the analysis of mRNA therapeutics. So this workflow enables direct RNA sequence identity and integrity analysis. So unlike often many RNA sequencing approaches, this is a direct analysis. There's no amplification step required. We can directly analyze our mRNA molecule of interest. So the workflow we developed is to use partial RNase T1 digestions by having them immobilized on magnetic beads. This allows us to control the reaction and stop the reaction. We then performed ion pair reverse phase chromatography for high resolution separation of the oligo fragments, combined with MS analysis on the Orbitrap Explorer 240 instrument. The sequence mapping was performed in a fully automated fashion within Biopharma Finder software using the MS2 fragmentation for oligonucleotide identifications. So using this automated workflow, this enabled us to identify over 80% sequence coverage of an RNA in less than 90 minutes in a fully automated fashion. I've shown here a link to some of the recent work that we just had published. So if they want to find some further information, then this was recently published in Analytical Chemistry, and the link's there for this particular publication. So with that, I'd like to conclude and just acknowledge a few people, in particular, Dr. Christina van Hinsberg in the group, who performed most of the development of the workflow and the data analysis, in conjunction with Dr. Caroline Evans and James Gunham in the group. And in particular, I'd really like to thank our colleagues and collaborators at Thermo Fisher Scientific, in particular, Ken Cook, Andrew Williamson, Angela Crisciulio, John Bardley, Jennifer Sutton, and Keely Murphy.
Thank you very much, Mark. I am sure you will all agree that there were some excellent points raised during the presentation. And now, before we start the Q&A session, we have our second poll question. Please answer by selecting any options that apply to you. The question is, what features of the LC-MS workflow do you find most useful to you? Fast, 90 minutes from sample to sequence map. No need of library generation. Comprehensive sequence coverage. Universal, works on both unmodified and modified RNAs. and reproducibility. I'll give you all a moment to submit your responses. Thank you. We will now begin the Q&A session. To submit a question to our speaker, you should enter your question into the box on the right-hand side of your screen and click Send. We will answer as many questions as possible during the time available. We've had several questions submitted already, so I'll move straight onto these. The first question is, do you quench the digest? Please explain more in detail the digestion with magnetic beads.
Thank you very much for the question. It's a really good question. In this case, we don't quench the digest. The big advantages of using the RNAs T1 immobilized on the beads is the ability to simply stop the reaction by removing the beads using essentially using magnets in an automated fashion. So by having the RNAs immobilized on the beads, we can simply stop the reaction at a defined time point by removing the beads, removing the enzyme from the reaction. So we don't have to worry about quenching the reaction and stopping it. This is a particular problem with trying to do it in solution, because trying to stop an RNA's activity is really difficult. If you're doing an in-solution digest, stopping the activity is really difficult. It tends to just carry on degrading your RNA, even when you add chemical things like inhibitors to stop the reaction. So the big advantages of the immobilized beads are control of the reaction. We can simply stop it. And also it stops any RNase contamination of your chromatography column as well. And we've been working with RNases for a long time. And what we found also with RNase enzymes is if you do an in-solution digest and inject that material on your chromatography column, the RNAs can remain active on the chromatography column and also digest your material. And again, that's not something we really want to happen in partial RNase digests.
Thank you. The next question that we have is, what is the role of HFIP in RPIP HPLC? Isn't the amine doing the ion pairing?
So that's another good question. There's a lot of complexity that surrounds the kind of the ion pair chromatography and the role of HFIP and the amines. So in this case, we're using, in traditional kind of ion pair chromatography, you would use the ion pair with the acetate as the counter ion. So something like triethyl ammonium acetate for your ion pair chromatography. However, the problem with that comes, you get quite a lot of signal suppression in the mass spectrometry using acetate as the counter ion. So you've got a number of options. One is you could add HFIP into the triethyl ammonium acetate to improve mass spectrometry sensitivity. Or the other option is, and the approach we've taken here, is to use the TEA HFIP, where HFIP acts as both a counter ion for the ion pair reagent, and also, again, you get the benefits from the improved ESI-MS sensitivity. So in this case, one of the advantages we also find is just having the TEA HFIP mobile phase is that our pH is a little bit higher than neutral pH, and this shifts our charge state to higher charge states where we get beneficial sequence coverage of fragmentation. So we're doing typically the ion pair chromatography around about pH 8, high pH 8, 9, and this has certain benefits for the analysis.
That's great, thank you. The next question is, could you use this method to identify chemical modifications on mRNA on specific bases?
Yep, so this workflow will allow you to identify chemical modifications on certain specific bases. So the example I showed you in the slide was where we generated or used an mRNA with all the uridines replaced with 5-methoxy-U or N1-methyl pseudouridine. So we're able to essentially use this approach to identify the known modifications in the sequence and allows you to do the sequence mapping. And we've also extended this to some of the work we're doing here in Sheffield, and that's looking for post-transcriptional modifications of RNA, such as ribosomal RNA. And you can use this same strategy again using the partial T1 digest in combination with the data analysis to identify a broad range of different chemical modifications of your RNA.
Thank you. The next question is, did you combine a partial RNase T1 and RNase A digest?
That's a very good question. It's not something we've showed in the data in this slide here today, but it's something we're working on at the moment, and we've got some data here in Sheffield. So yes, one option is to obviously just doing the partial T1 digest alone gives you really high sequence coverage, above 80% and above 90% in some cases. But of course, if you want to generate 100% sequence coverage, it may be we can use a combination of different enzymes, and that's one strategy that we're using at the moment. So combining partial T1 digests with partial A1 digests to give to combine those two data sets for comprehensive analysis.
Thank you. The next question is, have you tried using this on analyzing several RNAs at once? Although that would definitely have its challenges, could this be used for complex RNA mixtures in the future, or which would be the key challenges attempting that?
Yeah, that's a really great question. I showed some data on that very last slide where we just looked at 2 or 3 different RNAs in a sample mixture in different mass ratios. In that case, it was just the mRNA therapeutic of interest and the 16S and 23 ribosomal RNA from E. coli. We're just doing some further work now, extending that, looking at bigger mixtures. Of course, from a kind of biopharmaceutical analysis area, I know there's kind of interest in the field for mixing different types of vaccines, different types of mRNA vaccines together. So you've got multiple different mRNAs in the same mixture. So again, it should be feasible to analyze those. I think just the problem moving forward potentially is, as long as obviously we've got multiple RNAs, I think identification of those will be fine. But I think what you're going to generate is much more complex mixtures. So your sequence coverage is essentially going to just be reduced. So I think from an identification point of view, it's going to be possible to identify multiple RNAs. But of course, you're generating much more complex oligonucleotide samples. So the numbers that you may then identify in the sequence coverage will probably be reduced. So that's the key challenge, kind of maintaining high sequence coverage as the complexity of the sample gets bigger and bigger.
Thank you. The next question is, what is the advantage of this workflow compared to conventional next-generation sequencing, other than being able to identify mRNA modifications?
Okay, because obviously there's a number of RNA sequencing approaches and methods out there. I think some of the key highlights and the difference of this approach, first and foremost, it's a approach where we can directly analyze our RNA of interest. So there's no amplification required. We don't have to generate any next generation sequence libraries, etc. So we can analyze directly our RNA sequence of interest. I think the other big advantage is just the speed of the process. We've got it in a fully automated fashion now, where we can take our RNA sample, all the digestion's done in an automated robotic system, provides us a sample vial at the end, which then goes straight into the mass spectrometer. And then in a typical run, chromatography run, 40 minutes, data analysis. So combine all that together, the digestion, the LC-MS, and the data analysis. Typically, that only takes about 90 minutes to get your sequence identity and sequence confirmation. So I think they're the key advantages over alternative techniques such as next generation sequencing.
Thank you. The next question is there any fear of residual or carryover RNAs in the LC-MS system that causes unwanted background digestion? Okay,
so that's again a really good question, and kind of just addressed it before with the kind of the immobilized beads, which offer these big advantages of kind of contamination of the RNAs and the chromatography columns. So obviously we really want to keep these partial, longer oligonucleotide fragments intact. So what we certainly don't want is any residual degradation, either in the reaction mix or on the column chromatography. So by having the RNAs immobilized on the beads, we simply remove all of the RNAs after our time course, and then inject that onto the LC-MS system, where we don't see any RNase contamination in the chromatography column, and we're confident we've removed all of the RNAs, so we don't get any further digestion whilst it's in the sample vial.
Thank you. Another question here is, in terms of the last part of your talk, when it comes to impurity characterization, how did you address the problem of MS ionization difference in ssRNA sequences resulting from digestion and achieve reliable quantitation?
Okay. So for most of the analysis we performed here, it was essentially a qualitative analysis. So we're just looking to use the partial digest to generate a series of oligonucleotides, and then identify them, sequence them in the mass spectrometer. We weren't doing any quantification. However, it's true we do generate a broad range of different fragments, from small oligonucleotides down at 5 to 8 nucleotides in length, all the way up to some really large molecules, kind of 20, 30 nucleotides in length. And of course, there are ionization differences between them. So the bigger ones are obviously more challenging. The ionization fish is a little bit lower. But in this case, we're not doing anything quantitative. It's purely sequencing and qualitative analysis. I would say one of the difficulties of the large oligonucleotides is just ensuring we don't have any sodium and potassium adducts present, because the larger oligonucleotides tend to give you greater numbers of sodium and potassium adducts, which again can lower your ionization efficiency. So it's really key to make sure that use very high grade solvents to minimize the potassium and sodium adducts in your system. And you see these a lot more in these larger oligonucleotide fragments.
Thank you. We have time for one more question, which is, do you plan to study capping, percent cap 1 determination compared to uncapped cap O and cap G form with this method?
Yep, so we have used RNAs mapping approaches to look at the 5' capping efficiency, looking at the different forms. And obviously, there's a number of different strategies you could use there, depending on the sequence of your mRNA. So it's possible to use also complete digest, depending on the sequence, because that one might give you a defined oligonucleotide fragment. And then you can look at the amount of capped, uncapped material, again, so using the mass spectrometry workflow. Again, it's also possible to do this approach using the partial digest as well. So there's some advantages and disadvantages of each method. The partial digest method will give a number of different size fragments where you can see the capped and uncapped product, but it will allow you to also not have a problem with, depending on what the sequence is, where it may be too short, depending on what RNAs you're using for a complete digest. But the kind of the downsides for a partial digest is obviously you're going to potentially generate 4 or 5 different fragments that have the cap on, making quantification a little bit more difficult, because you have to sum up the different fragments with and without the cap on.
Thank you very much, Mark. That's all we'll have time for today, but just to remind everybody, any questions that we didn't have time to get to today will be answered offline as soon as possible. You can continue to ask questions even when watching this webinar on demand. A certificate of attendance is available to download from the Handouts tab on the right-hand side of the webinar platform. The webinar will now be made available on demand. If you have any friends or colleagues that you think may be interested in this webinar, please feel free to share the link. Thanks again for your time.